Welcome back to All About RX. Today we're bringing high energy, literally, and laser focus to demystify the electron transport chain and oxidative phosphorylation. In this explainer, we're going to break down exactly how our cells turn biochemical fuel into usable energy, and more importantly, we're going to show you how to absolutely crush the questions you'll see on your exams.
To make this as logical and testable as possible, we'll take a macro to micro journey today. We're starting with an energy harvesting overview, exploring the mitochondrial architecture, breaking down the 5 ETC complexes, mapping out site-specific inhibitors, and wrapping up with a quick exam recall.
All right, Section 1, Energy Harvesting Overview. Let's start with the big picture here. How do our cells actually harvest energy from the food we eat? Well, as energy-rich fuels like glucose, fatty acids, and amino acids are oxidized, they donate electrons to coenzymes like NAD+ and FAD. This forms NADH and FADH2, which basically act like cellular delivery trucks. They pass these electron pairs right down to a specialized set of carriers embedded in the inner mitochondrial membrane. And as those electrons move down that chain, the free energy that gets released is partially trapped as ATP. This incredibly vital process is known as oxidative phosphorylation. It's simply ATP synthesis chemically coupled to electron transport. Moving on to Section 2, the Mitochondrial Architecture. Now let's physically zoom in to understand exactly where this energy magic happens. The final common pathway for these electrons sits right here, deep in the inner mitochondrial membrane. It's surrounded by this gel-like interior called the matrix, which is absolutely packed with enzymes for the TCA cycle, pyruvate oxidation, and beta oxidation. Now, watch out for this. It's a highly testable concept for your exams. The outer membrane is freely permeable to most ions and small molecules, thanks to its pores. But the inner membrane, it is strictly locked down. It's completely impermeable to hydrogen ions, sodium, potassium, and ATP, meaning it requires very specialized carriers to get things across. To maximize its output, that inner membrane is folded into these structures called cristae, massively increasing the surface area. In fact, it houses about 50% of its proteins directly for electron transport and oxidative phosphorylation. And it also holds ATP synthase complexes that look like tiny spheres protruding straight into the matrix. Okay, Section 3, the 5 ETC complexes. With our spatial map established, let's dive into the distinct enzyme complexes that actually do the heavy lifting here. So the inner mitochondrial membrane houses an assembly of 5 separate enzyme complexes. Complexes I through IV handle the actual electron transport, while complex V uses the resulting proton gradient to catalyze the synthesis of ATP. Let's be really precise about where our electron donors enter. NADH drops its electrons at complex 1, also called NADH dehydrogenase, via bound FMN and iron-sulfur centers. FADH2, on the other hand, bypasses this first step completely and donates directly to complex 2, or succinate dehydrogenase. Fundamentally, free energy is released as electrons move from a reductant to an oxidant. They can transfer in a few different ways, maybe as hydride ions to NAD+, as hydrogen atoms to FMN or FAD, or just as single electrons to cytochromes. Basically, they're constantly shifting those component redox pairs along the way. So how does the actual flow work? Well, once they hit complexes I and II, the electrons are passed to the lipid-soluble mobile carrier Coenzyme Q, also known as ubiquinone. From there, they move to complex 3, the cytochrome BC1 complex, and then they're ferried by a second mobile carrier, cytochrome C, straight over to complex 4. Now, complex IV, or cytochrome A plus A3 oxidase, is super unique. It contains copper atoms and is literally the only carrier whose heme iron reacts directly with molecular oxygen. It combines electrons, O2, and free hydrogen to form water. Because of this, the ETC is actually our body's primary oxygen consumer. Crazy, right? On to Section 4, Site-Specific Inhibitors. All right, snap your attention here. This is the high-yield pharmacology roadblock sequence that is practically guaranteed to show up on your exam. These site-specific inhibitors violently block electron flow at precise mapped locations. When this happens, carriers before the blockade stay fully reduced because they literally can't pass electrons forward, and those after the block remain completely oxidized. You absolutely must memorize these pairings. Rotenone and amytal halt complex I,
Antimycin A stops complex 3, and cyanide, carbon monoxide, and azide fatally block complex 4. And remember, because electron transport and oxidative phosphorylation are so tightly coupled, blocking the chain immediately halts ATP synthesis.
Finally, Section 5, Exam Quick Recall. Let's rapidly synthesize everything we've built today into a single, high-yield summary.
Here is your high-yield checklist. First, location. The ETC sits in the inner mitochondrial membrane across 5 complexes. Second, your mobile carriers are Coenzyme Q and Cytochrome C. Third, molecular oxygen is the ultimate final electron acceptor, turning into water. And fourth, know your inhibitor hotspots, rotenone amytal at I, antimycin A at 3, and cyanide CO anazide at 4.
To wrap up, I want you to think about this biochemical disaster scenario. If complex 4 is completely blocked by cyanide, what exactly happens to the oxygen we breathe and the ATP we desperately need? Ponder that for our next explainer. Thanks for studying with AllAboutRx. Don't forget to hit subscribe and check the link in the bio for these high-yield notes. See you in the next lecture.